Hi, I'm Brooke Crowley. I'm an associate professor at the University of Cincinnati in Cincinnati, Ohio in the United States. Just to give you a little background about who I am to answer some of these questions, I'm a stable isotope ecologist and paleoecologist, and I've been working on projects related to Madagascar for over 15 years. And stable isotopes, particularly stable carbon isotopes, are very useful for addressing questions regarding grassy biomes because they can directly and unambiguously assess photosynthetic pathway. So for example, C3 versus C4 photosynthesis, and of particular grass species or other monocot species. And they can also help determine the degree to which particular herbivore species consume C3 versus C4 plants. And now the temporal window for these analyses is kind of limited to the recent past, so I can work from the late Pleistocene into the modern, but they can absolutely contribute to our understanding of the extent of C4 and C3 plants within a small geographic area, like the region around a subfossil site, or a larger region like the Central Highlands. And to date, what I've done is I've analyzed a lot of bones and fur from both modern and subfossil animals, mostly vertebrates, all vertebrates from the Central Highlands, and along with Lori Godfrey, Karen Sammons, and James Sanford, I just published the most complete data set available for both radiocarbon and stable carbon isotopes for subfossil vertebrates from the Central Highlands, and that's available in Proceedings for the Royal Society B. So that's a nice contribution. Some of those data were previously published and we collated them together, and others were new and hadn't previously been published. And of course there's still much we don't know, but these data do seem to suggest that vertebrates in the Central Highlands have mostly, but not exclusively, consumed C3 plants in the past, and that's still the case today. Now, a fundamental assumption of this work is that grasses that are found in the Central Highlands use the C4 photosynthetic pathway, and globally, most tropical and subtropical grasses do use C4. However, C3 grasses are able to outcompete C4 species in shaded environments, in moist environments, or at higher elevations, and those are the kinds of environments that we do see today in the Central Highlands and were possibly more prevalent in the past. And so this is something that we need to verify. So I'm currently in the process of confirming photosynthetic pathway for as many grasses as possible that we find in the Central Highlands. And these include both those that are found just in this region, those that are just found in Madagascar, and those that are more widespread. And this is a project that's in a collaboration with Heidi Schmidt at Missouri Botanic Gardens, Caroline Lehman, and Maria Vorontsova. Vorontsova. Sorry, Maria. In addition to verifying photosynthetic pathway, we're using carbon isotopes. Our team is also confirming curated herbarium specimens are indeed correctly identified. We're exploring how photosynthetic pathway may vary among species that likely have different spans of time that they've been present on Madagascar. And we're also investigating if there are any differences in photosynthetic pathway for fire and grazer adapted species. And so this brings me to the second question that was asked, in which we were asked to consider how our disciplinary understanding of grassy biome distribution or evolution is aided by cross-disciplinary engagement. So collaboration among researchers, in my opinion, is key, and particularly researchers with different expertise is key. And for example, I could never do what I do or the kind of project that I just outlined on my own. So I don't have expertise in grass ID. I'm also not a plant taxonomist, and this absolutely affects my research. So if a particular herbarium specimen is incorrectly identified, or if I think two species are closely related based on old taxonomy and that's been updated, this affects the kinds of results conclusions I draw from my results and the usefulness of my results for others. And so that's really important, and I want to make sure I'm doing things correctly and the things I publish are correct. So I'm very, very grateful to be able to work with others like Maria and Caroline, and I encourage others to collaborate as much as possible as well. And that's one of the things that's so exciting about this workshop, I'm really, really excited to be able to be here and to participate and be part of this with others. I think one of the biggest levels of disagreement among researchers so far has resulted from a lack of understanding about what data show, and I think that there are two primary reasons for this. First, it can be challenging, if not impossible, to fully and correctly interpret data if one does not have training or experience in that particular type of data. And second, we don't always do a good job of clearly presenting our results or our interpretations. So we really have to be willing to work with others, or at least ask authors about their work if we don't understand it, or if something doesn't quite jive with our own understanding or our own research. And that could be data or results or conclusions or interpretations. And we also can all do a much better job, or at least a little bit of a better job, in making sure we clearly present our own research and that what we present is factually correct. So it's definitely easy to make small errors, but if we don't catch those, they can be perpetuated. And that's just it's reality. But the better we can do with that, it'll help everyone. And we should also always make sure that we do provide our data so that others can double check our work. So again, I'm really excited about this workshop. I'm just going to answer one more question here. I know I'm out of time, but regarding that third question about limitations within and outside my field with respect to inferred Holocene grassy biome distributions, there are assumptions that we all have to make about how robust our data are. And I'm currently working to address some of these assumptions in my own field. For example, that grasses in central Madagascar do indeed rely on C4 photosynthesis. However, there are still limitations in what I can do. And in particular, our record of the past is quite temporally and taxonomically limited, and it's also geographically patchy. Moreover, the kinds of questions I can ask with isotopes are inherently broad. For example, I cannot address what specifically an individual consumed, or what types of plants specifically were present in the past. I can only do broad swaths such as how abundant C3 or C4 plants were and how much those were consumed by herbivores. I also cannot readily distinguish the prevalence of C3 trees and C3 grasses, or for that matter, C4 grasses from other monocots that also use C4 photosynthesis, like sedges or rushes, but also they grow in really different types of environments than grasses do, using carbon isotopes alone. So this is something that I'm hopeful I'll be able to address and explore more using carbon and nitrogen isotopes. Nitrogen tells us something distinct from carbon and may be able to tell us more about habitat and environmental conditions. There may also be additional types of data and analyses like pollen spectra from sediment cores, or even compound-specific chemical analyses of sediments that could complement and enhance what we can say about the past, but those are still going to be somewhat limited by those other factors I mentioned, such as temporal and geographic limitations. So that's my longer than 5-minute video, but I'm really excited, and I'm so happy to be part of this, and thank you very much for putting this together, and I look forward to interacting with everyone again soon.